Very much for inviting me to speak. Yes, I'm local, and I'm glad that you were able to make it during the snowing morning, though last week we had 70 degrees and it was a beautiful spring. We will switch gears a little bit, and I'll talk more about cells rather than stem cells. And as I was introduced, that my primary research is in post-traumatic osteoarthritis. I don't know how many orthopedic surgeons are here or people who are doing research on cartilage and connective tissue, but I hope that the talk will be of interest to the entire audience. So here just disclosures that the work which I'm going to present today has been funded by a number of industry and foundation, as well as NIH and my endowed chair. And most of the work that I'm doing in my laboratory is with human cadaveric cartilage from donors. It is a cartilage which we receive fresh within 24 hours of death, and it is normal by all the characterization. And one of the exclusion criteria is that donor had no documented history of joint disease. So we have a unique opportunity to use fresh donor cartilage of all ages, all spectrum of normal preclinical pathological changes which have not been clinically diagnosed. So as many of you know that more than 12% of prevalence of severe osteoarthritis is attributable to post-traumatic arthritis in the hip, knee, or ankle joints. It is probably more than 6 million people just in the United States who have signs of post-traumatic osteoarthritis. And the problem is that patients who present post-traumatic osteoarthritis usually much younger than those who have idiopathic or degenerative osteoarthritis, which again brings a huge issue for treatment choices. And it is very expensive because it has huge impact on financial impact and many people with disabilities, which is presented at much earlier ages. It is even more serious problem in people who are involved in active sport and athletes, professional athletes. As you can see, the prevalence is increased by almost 3 times, more than 3 times, and there's a number of injuries which are associated or which can lead to post-traumatic osteoarthritis, and those are meniscus tear, ACL tear, or collateral ligament tear. And majority of these patients are symptomatic. If you compare the number of, if you look at the epidemiology of post-traumatic osteoarthritis, you can see that with more impactful sport, and impactful I mean more professional sport, the epidemiology of post-traumatic osteoarthritis is greatly increased, and it is 5 to 12-fold increased risk of athletes to develop severe post-traumatic osteoarthritis. So what is the problem? The problem is that cartilage is one of the connective tissue, or one of the musculoskeletal tissue with the lowest ability to repair or regenerate. It has, as you may know, that the structure of cartilage, it has no blood supply, no lymph supply, no nerves, and that's why the supply of nutrients which would help to stimulate cartilage regeneration and repair is very limited, and it comes from external sources, and that represents one of the biggest problems in regenerative medicine and for cartilage repair. It's not the only problem when you think about cartilage repair. Cartilage is a very thin tissue. The thickness of cartilage in human joints varies from 2 millimeters in the ankle to up to 7 millimeters in patella. It has to resist compression. It has to have low coefficient of friction. And it should be able to distribute the load of the body weight. And it has to last for quite a long time. It is also important to remember that spontaneous repair fails to reproduce originate normal tissue, which means that it might also have problems with the integration with the host tissue, both the bone underneath or cartilage on the side. Furthermore, because of it doesn't have enough time to build this right structure, as if you take an example, newborn cartilage, and it has time to mature and develop zonal distribution to be able to withstand the load, regenerative cartilage does not have this time. And that represents more challenges for this either spontaneously repaired or regenerated cartilage, because not only the issue with the integration with the host tissues, but it becomes more susceptible to re-injury and to inflammatory environment which might be presented within the joint. So again, I hope that I convince you that to regenerate cartilage is a big issue and it's not an easy task. If we adopt the diamond model, which was developed for bone regeneration by the Department of Thomas Einhorn in Boston University, if you think about cartilage regeneration, factors that we have to consider, that underneath it should be able to integrate with the bone. It requires chondrogenic cells, whether it is stem cells, bone marrow cells, or stem cells coming from synovium. It might need to have scaffold, not necessarily, but it may. It probably requires active agents which would stimulate matrix production, because cartilage is a very unique tissue. It has only 5% cells of its volume, and the rest is water and the extracellular matrix. Again, we were talking about mechanical environment, so it should have proper mechanical environment in order to build the right structure of the tissue. And on top, it has to face a synovial environment, which might be hostile, it might be inflammatory, yet it most likely provides stem cells from synovium. It also might require chondrogenic scaffold and chondrogenic cells. So what are the treatment options nowadays for cartilage injury? Primarily those are surgical options. Debridement lavage, just to remove the fragments. Marrow stimulation, which is largely based on bone marrow stem cells, which are coming up to repair the cartilage. Osteochondral transfer technique, and those include both autograft and allograft. And there is a number of cell-based cartilage repair techniques which are widely used. Those are with use of autologous chondrocyte transplantation, the method which was discovered and characterized more than 20 years ago. There's another way of inducing cartilage repair by the use of characterized chondrocyte implantation, scaffold-associated chondrocyte implantation, and those are a number of different ones owned by different industry. Neocartilage transplantation, if people regenerate cartilage in vivo and then transplant the entire cartilage, and as well gene therapy and stem cells, when cells are either transfected with genes of interest or stem cells are used for cartilage regeneration. And of course, there's a number of biologic ideas which are now part of the clinical trials, and I'll get back to that. And those include platelet-rich plasma, growth factors, caspase inhibitors, and others, and cartilage tissue engineering. There are some emerging experimental approaches for cartilage repair, and those are based on the use of periosteum cells or use of physical methods to stimulate matrix production. Those also can be cellular or acellular scaffolds, hydrogels with or without cells, because I don't think that there is a uniformity in opinion that you absolutely necessarily need to bring the cells. Maybe there's enough cells to come from the bone marrow and from synovium, and nanoparticle technology as well. There's a lot of new developments on the use of therapeutic based on vitamin D, growth factors, and I'll touch upon those for a little bit, and specific pathway targeted therapy, and the list of those are here. So what I'm going to share with you today, a few studies that we have done in my laboratory, and those when we try to characterize chondrocytes used for autologous chondrocyte implantation, the methods which is introduced by Zimmer de novo using juvenile cartilage, osteochondral allografts. There's a new technology that we are very excited about, introduced by Cartihill company from Israel, and biologic approaches which we are widely using in my laboratory, trying to understand the mechanisms which drive cartilage regeneration in post-traumatic osteoarthritis. And those are experimental ones. So let's talk about autologous chondrocyte implantation. Probably all of you, or many of you, are familiar with this method when chondrocytes are taken from the normal cartilage, non-load bearing area, used in vitro for expansion, usually cultured for a couple of weeks, and then they transplanted back onto the periosteum into the defect in the joint. So what we did, we took these leftovers after the expansion, those which were just leftovers after surgery, and we wanted to understand the biology of these cells. So what we noted that, and usually chondrocytes are rounded cells, that's how they look in the monolayer cultures. As soon as we try to plate them and culture, they immediately change the phenotype, and they become more fibroblastic with elongated shape, which means that they would produce different matrices than primary chondrocytes. So we try to understand how these cells respond to stimulation with growth factors, as we always used in our models with primary cells. And what we found that with normal physiological doses of growth factors, whether it was osteogenic protein 1 or insulin-like growth factor, there was basically no response. These cells required larger doses or combination of these factors in order to produce at least proteoglycans, one of the major components of cartilage. We also found that if we put these cells in scaffold, they like it more. They do keep their phenotype, but without any stimulation, many of them are dying. So again, it seems that their survivorship is lower than primary chondrocytes. But when they're treated with these growth factors, they can expand and then they can perform quite okay. We went further. It's a busy slide, and I'll walk you through. So we looked at these scaffolds, and we wanted to see what kind of matrices these cells deposit, because it's very important to know that if you inject it into the patients, these cells deposit the right matrices. So here's this panel is normal chondrocytes, primary chondrocytes. And you can see that under combination with growth factor, especially they deposit well-defined collagen type 2 and aggregate matrices. While if you look at the same group in the ACI cells, you don't see as much matrices. You don't see this matrix to be deposited in these cells. They do respond to individual growth factors treatment, but much to lower extent, which suggests that these cells do have altered phenotype and their metabolic activity is changed compared to primary cells, which again brings the question how good this technique, and at least it can give us some ideas why in some patients there's failures which are noticed and documented. Another approach which is widely used by orthopedic surgeon is the use of allograft, osteochondral allografts, when the plugs of donors are inserted in the areas of the defect. Again, we wanted to look at these cells because we did hypothesize that when you impact or insert these plugs into the area of the defect, you subject these plugs to two big challenges. One, the impaction, like mechanical influence on the superficial layer of these cells. And these cells usually are inserted into kind of pro-inflammatory environment which comes from the injury and from the joint. So we mimicked this idea in the lab, and what we found that here's the superficial data for superficial layer, and here for the entire cartilage. Red is osteochondral allograft, and black is fresh. So what we found that at the beginning, it seems that both have relatively similar viability, whether in the superficial layer or throughout the entire depth. But as soon as you add insertion, because that's how, or impaction, which mimics this insertion, you can see that the viability of the superficial cells drastically decreased. And it's primarily in the osteochondral allograft cells, because these plaques or these cartilages have been stored for at least 2 to 3 weeks. And you can see that these red cells, it is dead cells, and green ones are alive cells, and you can see that there is more dead cells as you subject these plugs not only to impaction, but also to the effect of inflammatory mediators, which we identified in synovial fluid of patients. So what it suggests to us that osteochondral allografts, and you can see these red lines all over this graph, that they have lower chondrocyte viability than original normal tissue, which again presents a challenge for appropriate cartilage repair. We looked at these plugs with safranin O staining to see the structure of these cells and these cartilages, and we found that as soon as you put these osteochondral allograft sections into the culture, you can see the depletion of safranin O staining, which again shows that there is washout of major matrix components. And what is very striking and very disturbing, that if plaques were stored for more than 3 weeks, you can see total change in the morphology of the tissue. Surface becomes smooshy and soft, which convinces me that it's very unlikely that this cartilage will do well in the joint. So if you summarize data for osteochondral allografts, it seems that this tissue is less viable and more susceptible to the influence of catabolic mediators, whether it is biomechanical load or cytokines, that the way these plaques are inserted is very critical, because it may set the stage for allograft failure, and that if it's stored for a long time, it definitely presents a problem for cartilage regeneration. So let me show you the next technique. It's the novel technique developed by Zimmer, when they use juvenile cartilage minced into very, very tiny pieces. They put it in fibril glue and put in the area of the defect in patients. So what we find that, and we compared these cells with the dull chondrocytes that we get from donor cartilage, which is fresh. So what we find that you can probably see here, that cells migrate from tissue, outside of the tissue, both from juvenile cartilage as well as from adult cartilage. If you compare here, with the exception of the older donor who was 74 years old, the time of migration when cells are in culture and they start to migrate out of the tissue doesn't change or is not varied between juvenile or adult cartilage, as long as it is within 50 years of age. You do see high metabolic activity in terms of gene expression in these de novo juvenile cells compared to adult cartilage. If you look histologically, you find that there's more matrices deposited by older cartilages or chondrocytes than by juvenile. So again, we didn't see that much difference between this preparation and adult cartilage or adult chondrocytes, as long as they're within 50 years of age. And but we also found that there is no need to mince the cartilage into those tiny pieces, because we use much larger pieces and we found similar results. So summarizing the NOVA approach, the prep itself is very heterogeneous, and its viability and migratory activity varies between specimens. High migration of de novo and adult chondrocytes was observed when high serum present and fibrin glue, which again may not necessarily be conditions when those cells are implanted into the patient. There's no difference in terms of the time when cells start to migrate, as long as it is within 50 years of age. And there's no significant advantages have been found in our lab in terms of use of de novo cartilage versus adult. So the next technique which I want to introduce to you, which we are very, very excited about, it is based on corals, modified coral. It is a company named Carty-Hill, and they use this bifacial construct which has coral and on top hyaluronic acid, which is meant to be embedded into the osteochondral defect, cartilage defects. And the idea is that this bone face will attract MSCs from bone marrow cells, and the cartilage face will attract cells from the superficial layer of cartilage or from synovium. And the company showed very encouraging data with 6 months follow-up and 12 months in the next slide, when cells which had this osteochondral construct or coral construct, cartilage regeneration and bone regeneration was much more apparent than those which did not have this construct. And you can see with a micro-CT that bone regeneration was quite appreciable versus a control which didn't have that one. So the question which we ask in the laboratory, what is the mechanism? How does it work? And whether human cells will respond similarly to the animal cells, because experiments which I show, animal experiments were done with goats. So we prepared the cartilage explants in the shape of donuts, and we inserted the construct in the middle hole. So what we found that, yes, cells do migrate from the tissue. You can see all these green cells which are migrating from the tissue. You can see that we were able to identify cells within the pores of the construct. And what is interesting, histologically, we can see that cells are migrating into the construct and they're depositing extracellular matrix. Our new data show, which I'm not going to present today, that many of those cells are stem cells or stem cells from superficial layer, which really doing the job for cartilage regeneration. Because in the control, when there is no constructs, cells also migrate, but they migrate from the deep layer and then immediately become hypertrophic, and they do not move as far and they do not deposit extracellular matrix. And we also found that in the presence of these coils, there is more production of proteoglycans in both knee and ankle joints, and which is important because it can be used for a number of different pathologies and a number of different joints. We always compare knee and ankle in our work. And these cells produce more aggrecan and collagen than cells in the absence of the control. So again, summarizing this part, that it seems that it is a unique acellular scaffold which is capable of inducing regeneration, attracting cells from the bone marrow and from the synovium, that the tissue, regenerated tissue appears to resemble hyaline-like cartilage in both in animals and in human cells. There was no adverse effects noticed and noticeable, and there's clinical trials which are using this construct now. And it also showed that it seems that this approach stimulates cartilage-specific gene production. And the people who are involved in this work truly believe that it might have strong evidence for clinical application. So now we're switching to post-traumatic osteoarthritis and trauma when trauma is inducing cartilage regeneration. So analyzing the work of multiple laboratories and using our model when we impact cartilage like a dashboard injury, we were able to characterize cellular responses which specific for immediate cellular responses, which are specific for cartilage injury. And as you can see that there's about 3 phases of these responses. Phase 1, or early phase, which is characterized by cell death, both necrosis and apoptosis, elevation of pro-inflammatory mediators because cells are damaged and all intracellular components are released. Then there is some initial anabolic response, which is characterized by elevation of growth factors, superficial zone protein. And then there is kind of an intermediate phase of equilibrium between catabolism and anabolism, and then a remodeling phase, which is more characterized by anabolic responses. So based on these data, we decided to use a number of different therapeutics to see whether we can use mechanism-based approach to arrest cartilage regeneration. And based on these data, there are 4 key principles for therapy which can be used. Chondroprotection, to protect cells from death, anti-inflammatory, matrix protection, and anabolism, to stimulate matrix production. So in terms of chondroprotection, I'll show you data with 2 different treatments. One is we used P188 surfactant. It's just a regular surfactant detergent, which is very cheap, $8, very cheap from Sigma, like up to $10 for the prep. And what we did in this impaction model, what we found that if you impact the cartilage, of course cells die under the area which was subjected to the impaction. But what is more important that even the areas which were not subjected to the impaction, but they're adjacent to the impaction area, cells start dying regardless that they did not experience the impact, because those from the impact, they release cellular components which mitigate the cell death and expand cell death both horizontally and longitudinally. So when we applied this surfactant 1P188, we were able to arrest this spread of cell death both in the areas which were subjected to the impact, red line here, core, or in the areas which were adjacent and did not experience the impact. So at least we were able to show in the experimental approaches, both in the lab, and there were work on rabbits in other lab, which showed that, yes, you can arrest cell death, and thus you can prevent cartilage, or at least reduce cartilage degeneration induced by trauma. In the laboratory of Iowa Orthopedics, Dr. Jody Buckwalter lab, they use reactive oxygen species antioxidants, rotenone in this case, to see whether they can arrest cell death and cartilage degeneration. And you can see here all red cells after impaction because of cell death. And when they used rotenone, cell death was drastically reduced. We also found that regardless what kind of mechanism you target, it is possible to arrest cell death. So this line is normal, here impacted, and everything colorful is different approaches that we tested. So all of them were able to, a certain degree, reduce cell death. So the next question is it sufficient? Because cell death is only step number one, but is it sufficient in order to stimulate matrix production and reduce cartilage regeneration? So there's a number of growth factor families which has been considered for cartilage regeneration. I'm not going to go through all of them. I'll talk just about one, which was heavily studied in my laboratory for more than 20 years now. And this is bone morphogenetic protein 7 or osteogenic protein 1. And then I'll mention at the end what kind of clinical trials are now registered for cartilage regeneration. So what we found in our lab and in many labs throughout the country, that BMP7 is able to stimulate have a reparative effect in different models, both human and animals. Mosaicplasty, osteochondral defect, chondral defect. It also has protective effect in models, as I just mentioned to you, ACL injury and acute trauma. And all the mechanisms of its effect have been characterized. It has anti-apoptotic effect. It has effect on stimulation of matrix synthesis, on inhibition of key pro-inflammatory mediators like MMP13 production, and caspase inhibition. So it has a very wide, not only pro-anabolic, but anti-catabolic effect. In our trauma model, in collaboration with Dr. Hertrick from Canada, we also found that within first week, if you apply BMP7 right at the time of injury, that we were able to, reduce apoptosis, we were able to protect more cartilage, and to have a reduction in leukocytes. So in this particular case, BMP7 was injected at the time of injury and 1 week later. Is the effect sustainable? And what is the right timing for therapeutic application? So what we found that if the treatment is applied immediately or soon after injury, in this case 1 week, within 1 week, here 3 weeks later, and here 3 months later, you can see that 3 months later there was no effect. Growth factor was not able to arrest cartilage regeneration, yet when it was 3 weeks and specifically 1 week, it did have protective and regenerative effect. And the more point I'm trying to make that it's not only what kind of therapy, but the timing of therapy is absolutely crucial when you treat patients after trauma. So here's a list of clinical trials which aim to address different biologic approaches to cartilage repair. As I mentioned, de novo techniques, microfracture, which uses stem cells, autogenic stem cells. There's a number of studies with chondrocytes which are transfected with TGF-beta, another growth factor which is used for cartilage regeneration. Again, comparison of ACI and microfracture, neocartilage, again microfracture, platelet-rich plasma. There is more and more interest in plasma-rich plasma, in platelet-rich plasma, stem cell therapy, allografts, MSCs, matrix autologous chondrocyte implantation, and FGF18, another growth factor which has been tested for osteoarthritis patient, and that clinical trial basically failed because in degenerative late stage osteoarthritis, the growth factor FGF18 was not effective, and now it's a new trial which we haven't seen the data yet for post-traumatic osteoarthritis, when they really limited the age of patients and the pathology these patients present. So as you can see, it's not as much as one would expect therapies and therapeutic approaches to osteoarthritis, but basically it is cells, whether stem cells or different techniques to cartilage repair. So to summarize, what is the emerging therapeutic approaches for cartilage intervention in post-traumatic osteoarthritis? Mechanism-based to arrest cell death, to arrest apoptosis, antioxidants, anti-inflammatory, again, targeting specific pathway. There are some natural products which are becoming more popular now, resveratrol, green tea, polyphenol, vitamin D, and others. There is matrix protection, again, to inhibit matrix metalloproteinases, so other factor which can induce cartilage damage. And there's pathway-specific targeting opportunities, and anabolic, which is growth factor, small molecules, and platelet-rich plasma. Probably the most potent are those which have combination of different factors with different mode of action. And there's also on the market synthetic biolubricants, which are going to be tested for cartilage repair. Again, as I mentioned, it's very critical, the mechanism, time, dose, target, and treatment regimens. And if you use biological molecules, it's always important to remember that chondrocytes produce these molecules. So in the assessment of doses, one needs to think how much endogenous growth factor or how much endogenous biological factor have been produced by chondrocytes. With that, I'll stop, and thank you very much. And that's how Chicago looks at night without the snow.
We're going to have time maybe for one question in the interest of time.
Here you go, ma'am, right here.
Thank you. I would like to ask, have you did a comparison between mesenchymal stem cell and chondrocyte autologous for clinical trial for chondrous repair for cartilage?
We haven't done direct comparison. We don't work that much with MSCs, but there's a lot of literature on MSCs, and these cartilages, which, I mean, the data which show with MSC that cartilage which was generated or regenerated, it doesn't have the right structure. It has the same problems with integration, with the host tissue, sustainability, and being able to sustain the load. I mean, it's all the same problems as I was talking about cartilage regenerated from different means. I haven't seen absolutely convincing data with MSCs that, yes, this cartilage looks perfect. And again, the question is whether it has to look perfect, whether even if it is fiber cartilage, but it can delay the time for surgical intervention, is it going to be good enough? And I don't think that we are ready to answer this question as well.
Okay, thank you.
You want to make a quick question? Okay, just one.
I was just wondering if maybe a tissue engineering approach for inducing the mechanotransduction and maybe selecting for cartilage that actually were more load-bearing is something that that you think might be the missing link in getting to the point where you get the proper kinds of cartilage production from MSCs.
You mean, I'm not sure that I understood what you meant with load-bearing versus non-load-bearing.
Right. So I mean, you know how muscle tissue that's been reconstituted and reconstituted into tissue in vitro approach has to be actually exercised in order to get it to develop into functional muscle tissue. We tested chondrocytes from both load-bearing area and non-load-bearing area. As soon as you remove chondrocytes from matrix, they lose this spatial specificity. If you keep it as explants, you might, but again, when you transplant it into the joint, it's a whole different scenario. So once you start manipulating them in vitro, there's not much difference in terms of sensing biomechanical load and responses to biomechanical load. Thank you.
Thank you very much.